We've got our next speaker, Caspar Barnes. Caspar is the founder of a New York-based decentralized biobanking startup. His educational history, when I saw it, I was like, wow, that is so diverse and perfect for a startup. Basically, he started in economics and neuroscience, moved over to business school, and then did his master's in biotechnology. So very much suited for a biotech startup, I think. Here he is.
Awesome. Yeah, thank you so much for the introduction. It's a real honor and privilege to be here. And I'm super excited today to introduce perhaps a new concept within DeSci. You've heard of funding, you've heard of publishing. Now it's time to talk about biobanking and the journey of donated biosamples. So my name is Caspar. I'm the founder and CEO of a startup called Amino Chain. And what we're building is the first decentralized biobank for enterprise healthcare institutions. So first and foremost, the experience for a patient that is donating a cancer biopsy sample today is a one-way street. A researcher comes up to them and says, hey, can we have your sample? Here's a consent document. Sign it, take the sample, the two part ways, and they never see each other again. So we put together this tiny little schematic, but as you can see, you have the patient on the left. They take the samples, and they give the samples and distribute them amongst an industry of pharmaceutical companies and scientists and researchers. They create the scientific upside and the monetary upside, and none of that is ever distributed back to the people that make the research possible. As a result, we have rampant mistrust within scientific research, and that's a notion that we know absolutely everywhere. So consent rates are as low as 25% at some major institutions. The people that do not consent to having their donated samples and data used for scientific research are patients of color and marginalized communities more often than not. So that is a very, very big issue that affects everything downstream. If we only have homogeneous samples and data, we only do research on diseases that affect people of Caucasian descent, which means we only create drugs for people that are affected within Caucasian populations. So we desperately need to build better ways to engage people within scientific research. The next problem is, after that, a sample will be taken from a patient. It'll be used within a scientific study once, and then after that, it's going to be put into something called a biobank, and it's going to be cryopreserved there for a long time. So what is a biobank, you might ask? Well, a biobank is a really big fridge full of donated human samples, and biobanks are fantastic because they catalog different diseases, and they standardize research output, and they operate at this intersection of research, clinical care, and informatics. So they're a powerhouse of precision medicine, if you will, and they've been growing in immense popularity from the 90s until today, coupled with an abundance of genomic information. So we love biobanks. They're fantastic. They're going to keep growing at this crazy rate, but biobanks have a number of issues. The first one is that on average, they'll only use 10% of the samples that they have stored within the inventory, and the average biobank of around 50,000 samples has $40 million worth of material that they're either not using themselves or not distributing to other people. So in the United States, that means that we have around 200 million high-quality samples locked up across all these biobanks. People don't know who has what, and trying to acquire those via complex licensing agreements called MTAs can take up to 8 weeks and is a process that every single biobank hates doing. So the kind of picture that we end up with within human subjects biomedical research right now is that all of these high-quality samples are locked up, they're hard to access, and the ones that we do have are very homogeneous backgrounds. So the solution to this is something called decentralized biobanking. How it works is every biobank installs this node software that we've created. It's something that they run on their servers. It integrates into their endogenous tech stack, like their lab inventory management systems, data capture softwares, EMRs, whatever they use at that biobank. And it abstracts away this blockchain protocol and connects them with all the other biobanks and pharma companies and hospitals running a node in the network. Within this network, a biobank can open up their inventory to a network of collaborators. Any one of these collaborators can streamline licensing agreements, taking samples of each other's shelves. And the best part of all of this is that the patient that donated those samples can track where they go within the decentralized system. They can earn royalties whenever their samples are transacted upon. And they can stay connected for translational therapeutics, for clinical trial enrollment, or for real-world data management. And so if the video is ready, we can show a quick demo of the system that we built. Awesome. Okay, so I'm going to have to look backwards here to explain this. But what you're looking at over here, could you pause it really quickly? Yeah, okay. So what you're looking at over here is something called Open Specimen. Open Specimen is one of the main LIMS vendors, lab inventory management software. So this stack is used currently at the Columbia Biobank. It's used at the Hopkins one. It's used at Stanford and so on. This is the bread and butter of tracking what's happening with your biosamples at a given biobank. So researchers are currently using this today, and we can show you as part of this demo, I'm now a scientist in the lab logging the fact that I've just received a new donated biosample. So if you can click play. I would come here and say I've just received whole blood. It's around 200 milliliters. It's non-malignant or whatever other data you want to put in about the sample taken from the abdomen. You add a barcode or a label, so you de-identify the sample, and then click Save. So now the blood is in the fridge, and our node software scans for updates every minute, finds that a new sample has been stored, mints an NFT out of that sample, and holds it within the wallet at the node address. So through this system, the biobank now has 50,000 samples in their fridge and 50,000 NFTs that represent those samples. After that's been minted, the donor of that sample comes and logs in with their email address, and this is what they see. They've done 4 donations, different times and different amounts. When they go to the donations tab at the top, they get the high-level overview, 4 donated samples, no studies happening just yet, how many lives impacted. So back to the donations tab. They click on the sample that they just donated, and then they get a little bit of information on the specimen itself. They can view the technical details about when the sample was minted, and they can see where the sample is and how it's being used. So for example, this one was just donated to the Columbia Biobank. So that's pretty dope, 168th Street. So yeah, that's the experience for the patient. Now, obviously, that NFT is also going to be put onto a decentralized biosample marketplace. So this biobank, Columbia, opens up the sample to a network of collaborators. So now we're logged on as a pharma company, and we want to see who has what samples where. We can see on our providers tab, in the network right now, we can check how many biobanks there are. Okay, that's pretty cool. There's 7 biobanks all running a node in the network. I can request access to see their inventory. But right now, only 1 has granted me access. So we can see here, this biobank, Advancing Site Network. You can check out their profile page. They seem like a pretty legit biobank. Access granted, that's pretty cool. So we can go to our view of the specimen center, which is our marketplace, and look for samples across all the biobanks that have given us access. We can filter by type, by quantity, donor information, by pathology of the samples, whatever we need. We then have this cool Airbnb type filtering thing. We can look for the samples that we need, filter by pathology if we have to. And then what we have at the end is all of the samples that fit our criteria, and these are NFTs. So if I click on any one of these NFTs or these samples, all this data that I get about the biospecimen is pulled from the inventory management systems and data capture softwares of the biobank. So this is automatically updated in real time relative to how they use the sample there. I can check out other samples from the same donor, add it to my cart, and then I can go and request the specimen. The whole view, actually there's one other thing to mention, is you can add additional services for things you might need to have done on the sample. Maybe we want some OCT imaging done, add a quick message for this biobank, and then we can submit the request, which is about to happen now. So the main point here is that we essentially are trying to turn all of these biobanks that have all of this high quality inventory of biosamples locked up in different places into shoppable specimen center experiences. If you think about a sort of Amazon of biosamples where you really try to distribute them as much as possible, and you have one interface where you can see who has what, see all the data you need about those samples, and acquire and license them in 5 clicks. That's the kind of unlock that we want. That's the kind of experience that we want for researchers, biobanks, and biopharma companies. The next flow that would have happened over there within the demo would have been, it moves towards a licensing agreement. You have ways where you can upload a template, you can redline clauses, and then you can sign and execute the agreement. Digital signatures manage the execution of that licensing document. Once the master licensing agreement is in place, you can just take stuff off each other's shelves. You don't have to do a licensing agreement each time again. And that is a huge value add for these biobanks, because on average, they'll take around 2 months to execute one licensing agreement today. So that is the system as it exists right now. But what is the real big vision that we're going after here with Amino Chain? The idea is we're trying to create a circular biobanking operating system within human subjects biomedical research. What that means is circular economies for people partaking within scientific research. So today, if the experience, you give the sample and then they have the value, and I see none of it, of course we end up with a system where the only people donating are purely altruistic. And let's be honest, being purely altruistic is something that comes from a position of privilege, right? You don't have to think about people that you're trying to feed. You're not trying to put food on the table. You're not working 10 jobs. So you can think about donating blood, donating stem cells, or partaking within a scientific study. So what we really need is bigger or higher incentives and a better system of transparency within human subjects research. And that's kind of what we're trying to create here. If you can give personalized consent for how your samples will be used, you have a marketplace that distributes it to everyone that upholds the consent. You can track the value of it the entire time, and value accrued goes back to you or your community in the form of a DAO or in the form of an individual. That's an incredibly powerful notion that means that we can have sustainable ways and sustainable economies for scientific research. The really cool thing as well is that we can build applications and economies that people can't really fathom right now. And that's what gets us really excited within the world of DeSci, right? If you try to explain to a researcher right now, you'd be in the lab holding a de-identified sample. You find out, it has an HLA polymorphism. You think this is a predisposition towards MS or an autoimmune disease, but you're not sure. You can push one button and a ZK bridge back to the medical records of the donor that donated that sample can return a proof saying, yes, they've been diagnosed with MS, or no, they haven't. That's real-world evidence that greatly informs the value of your research without compromising GDPR or compromising HIPAA standards. Equally, if you then create a drug that targets this polymorphism, you can just click one button, broadcast a message back to the donor, and ask them to be enrolled in a trial. And so all of these things can be massively automated and massively streamlined in a way that people can't exactly predict right now. And that's kind of why we're all here in this room trying to realize a future that scientists don't quite understand just yet. So with this distributed node architecture, our whole vision is we would love to collaborate with other DeSci projects. We'd love to find ways in which this architecture can be useful for others. This could easily spin into the world of publishing, into the world of financing. And that's the beauty of blockchain, right? The composable systems for scientific research. So on that, say thank you very much. I don't have a cool QR code, but I have an email, so. Yeah? Okay. I've just been given the all clear that I can take some questions in case anyone will ask questions. Yes?
Yeah. And compliance in general, could you talk a bit more about that? And the second quick question is, have you really gone out of the building and talked to all these biobanks? Is it being implemented? What was the feedback?
Yeah, absolutely. Okay, so first thing on the compliance and regulation standard. As we mentioned over there at the beginning, there's 200 million high-quality samples locked up in different places. At least in the United States, if you de-identify those samples, then it doesn't count as human subjects biomedical research. So you don't need consent again to distribute them, right? Terrible regulation. All that means is remove 18 identifiable, like your name, your email address, and your social security, and then you can put this stuff on a marketplace. So that's like the lowest hanging fruit right now, right? Every biobank has this backlog of inventory they'd like to distribute to others. We can go to all of them and say, we'll put them into one interface. We'll go to the pharma companies and advertise that for you, and we'll get those things distributed. The next step thereafter is what you call prospective collection. And that's a system in which you have biotechs or pharma companies saying, here's my SOP, I need you to get me a fresh set of breast cancer samples and harvest them according to this process. That requires a new consenting process. Patients have to know about this whole system. and it has a specific economy where even the doctors involved get paid for that extraction and movement, right? So that's what we want to build afterwards. And we want to have dedicated interfaces for consenting, for having IRB protocols approved, and then still having ways for the patient to be able to track where that sample goes, right? And the idea is that even if the sample is de-identified according to HIPAA or GDPR, afterwards, for secondary use, the donor can still see where their sample goes. So we can build on top of existing de-identification standards, where the researcher will hold a barcoded sample, but the patient knows that that barcoded sample belongs to them, as opposed to the researcher knowing it belongs to that patient. And so that's kind of where we're thinking in that direction. And yeah, we've spoken with 200 biobanks over the last 2 years. We've been to biobanking conferences. I was a keynote speaker at a biobanking conference earlier this year in Australia. And we had a really, really, really cool moment where the penny finally dropped with blockchain and how it's valuable for them. I had to give this presentation, try to explain what it was, demystify the technology, and then had to have this narrative of it's here to stay. Don't be scared of it. Embrace it. It's going to help you in so many ways. And I was explaining to one person how it worked. And in the meantime, another biobanker came over and it started to take over my explanation to them. So we started a little bit of a fire within the biobanking community, and people are getting excited about it now. It's like a really, really good time to keep growing this marketplace. And where we're at right now is 3 live biobanks, 50,000 samples, 170 biopharma procurers, including top 5 pharma companies and the NIH and a bunch of others. And yeah, now just growing towards hospital systems in Canada and on the West Coast.
Yeah, just one thing that wasn't clear to me because I don't know anything about biobanks. But can a sample just be used once, or can it be used a few times?
Can be used a few times. It depends on what the sample is, right? So if you create a standardized model, if you create an organoid, you create a cell line, that can be used many, many, many times over, right? If you take a breast cancer sample, you can turn that into certain slides. Those slides can be used a number of times over. If you take a genomic sample, that would normally get turned into a data set, and then that can be used many times over. The one things that cannot be used, like categorically can only be used once, would typically be things that you cryopreserve, right? That would be things like hematopoietic stem cells, allogeneic material, sperm and eggs. They're like those, I mean, you could maybe use them more for different cycles and so on, but those things would only be used one time, and they also cannot be cryopreserved, thawed, and then re-cryopreserved and then reused because the viability goes down.
Cool, thanks.
Do we have any more questions? I've got one, which is, so you were speaking about like the penny dropping and blockchain, and obviously this is sort of a Web3 native sort of idea. But when you're sourcing from patients, obviously a huge patient population has no knowledge of Web3 or really interest in it. Have you found that you've needed to make a lift to educate people about that, or can you kind of just abstract away from the whole concept and not really explain the back end of it? It's an NFT and all of that.
Yeah, the real value, I think, with all of us, literally all of us, is that we don't want to explain how the blockchain works. We just want to build applications that blows people's minds, right? That's kind of the game that we're all playing here. So that's exactly the same for us. If we can make sure that, I'm not sure if you would have seen with the donor portal when they log in, there's no like MetaMask connection and they have to get a wallet and handle their own keys. It's just sign in with your email, check out what's going on with your samples. That we got really good feedback from. We, like in New York, went to a few community health fairs in the Bronx and in Brooklyn, and we spoke with people there, and they were like, this would be phenomenal because I never know what's going on, and I never get any information, and none of it's tailored towards me and my experience. And so that's kind of the game that we're playing here. If we can work with the people building on account abstraction and bridging away all of the complexities of blockchain, it's the same way we don't really care how a Mac computer works. We just want the thing to work, right? So we have to build the same experience within crypto, and I think we're at the very early stages of that, but we're definitely doing exactly the things we need to be doing to get the adoption we want within like 5 years, 10 years or whatever.
Amazing. Any more questions? Cool. Okay.
Sweet. Thank you so much.